Hello and welcome to today's read-along. A read-along is a wonderful place where I read a text from right here in your screen, and you read the text along with me from wherever you happen to be. Our text for today is an article by Jake L. Weissman, Hal H.U., and Philip L.F. Johnson titled, "What Bacteria Do When They Get Sick." The online site Frontiers for Young Minds published this article under a Creative Commons Attribution license on July 24th, 2019. If you are interested in science, how our bodies fight sickness, and particularly how the biology of tiny creatures who get sick might help us be able to change DNA, you might like this one. Most people should be able to read this text with ease by the time they get to about 4th grade. That's about 9 to 10 years old in the U.S. educational system. If you are older than 10 and you don't find today's text easy to read, that's okay. Don't stress about it. But you should probably practice reading with a few read-along texts that are easier than this one before you come back and give this another try. Some suggestions for easier read-along texts are in the description down below. As always, you can also find a link to the text for today in the description down below too. So go ahead and find that link, click it, find the text so that we can read together. I'll wait. You got it? All right, good. So you can see that this is divided into sections, right? I will read the section headings, but I'm not going to announce when I change paragraphs. Do you see pictures called figures? I'm not going to read those or the captions, but they're really interesting, so you should go back and read them when we're done reading this paper together. I will read the abstract at the beginning though, and that's a summary of what the article is about. Scientific papers generally have an abstract you can read quickly to see if the paper is about something you want to know. Are you ready? Let's read! Abstract. Bacteria are really small, so small you cannot even see them. Did you know that bacteria can still get sick, just like us? In fact, most viruses in the world infect bacteria, not people. What happens when a bacterium gets infected by a virus? Well, just like us, many bacteria have their own immune systems that help defend against infections. Recently, scientists have discovered some new bacterial immune systems. We have been able to repurpose these systems for entirely new uses. One such immune system, called CRISPR, can potentially allow us to rewrite DNA any way we want in any living thing. Now scientists are using bacterial immune systems as powerful tools to precisely edit the DNA of all sorts of living things. Viruses infect bacteria. Did you know that bacteria can get sick just like you? If you have ever caught a cold or had the flu, you know it is no fun getting infected with a virus. Well, it turns out that most of the viruses in the world infect bacteria instead of people. Scientists call these viruses bacteriophages, which literally means bacteria eaters. There are around 10 to the 30th viruses in the ocean. That's a 1 with 30 zeros behind it. That is more viruses than there are stars in the universe. Most of these ocean viruses infect bacteria. Maybe all of this is new to you, but bacteria and viruses have been around for a very long time. Bacteria have been evolving alongside viruses since the origin of life. They have been locked in a constant battle for over 3 billion years. After all that time, bacteria have evolved a few tricks to defend themselves. Bacteria defend themselves just like we do. Your body has lots of ways to keep you from getting sick or to help you get better more quickly when you do get sick. Your first line of defense is your skin and the membranes inside your body. These keep nasty bacteria and viruses away, just like a wall. When you get a cut, why do you have to be careful to keep it clean? So that you do not get an infection. Sometimes, though, your skin is not enough and you do get sick. When you get a fever, that is a sign that your body is trying to fight whatever is causing you to be sick. There are some clever ways your body can fight infection. After your body fights off an infection by a virus for the first time, it can form a memory of what that virus looks like. That way, you would not get sick from that virus again. You will recognize the bad guy and fight back. We call this memory adaptive immunity, and it is why people usually only get the chickenpox once, and it is also why vaccines work. A vaccine shows your body a little bit of a dead or weakened virus, so your body can remember the virus and fight against the alive version later on. Some viruses change over time, though, so that these memories do not work forever. Have you gotten a cold or the flu more than once? That is because these viruses change quickly. Each time you get sick, it is actually a slightly different version of the cold or flu. Bacteria seem a lot simpler than us. After all, they are really tiny and only made up of a single cell. Bacteria do not have brains or other organs. Even their one cell looks much simpler than one of our own cells. Even so, bacteria can defend themselves from viruses a lot like we do. First of all, each bacterium is surrounded by a cell membrane and a cell wall. These structures are like shields that protect bacteria from the world, similar to the way your skin protects you. Viruses need to attach to the outside of a cell and poke through to get inside. If the bacteria change the shape of the cell walls, this can prevent viruses from sticking to them. Then the bacteria are protected from infection. What happens if a virus does get through a bacterium's cell wall? Well, some bacteria also have adaptive immunity, just like we do. That means that they can store a memory of a virus to help them protect themselves later on. Scientists only discovered this relatively recently. Before, nobody thought bacteria were complex enough to have something like adaptive immunity. Nature continues to surprise scientists with new, weird stuff. We call the system that provides adaptive immunity in bacteria the CRISPR system. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a complicated way of describing what CRISPR looks like. When scientists sequence the genetic code of some bacteria, their DNA, they found a pattern in which the same short sequence was repeated over and over again with some gaps in between, regularly interspaced. These short repeated bits were palindromes, meaning that they looked the same going forward or backward, like the words kayak and racecar. Finally, all of the repeats were found clustered close to each other in the bacterial DNA. Hence, the scientists came up with the name CRISPR. We admit that the name is pretty confusing, but I would not worry about it if I were you. Scientists do an okay job at figuring out how the world works. We are not great at coming up with simple names for things. How does CRISPR work? What do we mean when we say that CRISPR helps bacteria remember a virus? Well, to understand that, first you have to understand what a virus is. Unlike humans and unlike bacteria, viruses are not made of cells. Instead, a virus is a bunch of DNA or RNA, a molecule related to DNA, packaged tightly into a capsule made of proteins. You can think of DNA as a long book describing what a living thing is supposed to be like. For example, your DNA describes how your body should work, and this code, along with the world around you, shapes how you grow. All living things store their genetic code as DNA, but some viruses have RNA instead. RNA does pretty much the same thing as DNA for viruses, but is built a little differently. Think of the code for those viruses being written on stone tablets instead of paper. At the beginning of an infection, the virus's DNA or RNA gets injected into a cell. a human cell if the virus infects humans, or a bacterial cell if the virus infects bacteria. Afterward, the virus reprograms the cell. It makes lots of copies of the virus. Eventually, these copies are packaged into new capsules. Finally, the cell is broken open and lots of newborn viruses go off into the world to infect new cells. You can see how this works in Figure 1. CRISPR in bacterial cells works in 2 steps. First, when a bacterium has a CRISPR system, it can store little pieces of viral DNA. Each one of these pieces can be thought of as a different memory. Now the bacterium knows part of the virus's code and can recognize it as a bad guy later on. If a virus injects itself into a cell and that virus matches one of the bacterium's stored memories, then the bacterium knows that there is something wrong. Once the bacterium knows that it is being infected, it starts the second step of CRISPR immunity. The bacterium uses CRISPR to chop up the virus before the virus has a chance to make copies of itself. Voila! No infection. We show how this works in Figure 2. Of course, bacteria do not think or know things like we do because they do not have brains. All of the steps in CRISPR immunity happen automatically. But it is helpful to think of them as memory when trying to understand how they work. Why do we care so much about CRISPR? Recently, CRISPR's been in the news a lot. People are very excited about this odd little system that bacteria use to fight viruses. Why so much excitement, you ask? Well, it turns out that we humans are pretty good at stealing the tools bacteria have evolved over billions of years and using them for our own purposes. Scientists have been able to turn CRISPR into a way of DNA editing. Bacteria use CRISPR to cut the DNA of viruses in a very specific way. Scientists have figured out how to use CRISPR to cut any DNA very precisely any way we want. Why do scientists want to edit DNA anyway? Some human diseases are very difficult to cure because they're coded into our DNA. These diseases are not caused by viruses or by bacteria. Some examples are sickle cell anemia and cystic fibrosis. These diseases are caused by tiny changes to the genetic code. These changes make the body work slightly differently from the way it otherwise would. With CRISPR, we can edit DNA and maybe fix these small changes. This means that CRISPR could be used to help cure some of these genetic diseases. That is not all, though. Scientists are using CRISPR to design hardier crops for us to eat, to try to get rid of disease-carrying mosquitoes, and much more. We are still learning how to best use CRISPR as a tool. It is important that we make sure to use CRISPR for good reasons. Recently, a scientist reported that he had used CRISPR to edit the DNA of 2 children. He was trying to make them resistant to certain diseases. The global scientific community was very concerned when they learned this. Many scientists were worried about the scientific procedures used. Others doubted whether the researcher had acted ethically. Many scientists thought that the benefit did not outweigh the risks. The diseases that the edits would have prevented are easily preventable with other methods. CRISPR technology is not perfect yet and could be harmful. Often, CRISPR can introduce changes into DNA where we do not want it to. Using the technology on humans can be risky. It is important to remember that just because a scientist can do something does not mean they should. Scientists around the world frequently hold big meetings to discuss these issues and the best way forward. At those meetings, they ask each other and members of the public questions like, is it ever okay to edit a human's DNA? And if so, when would it be okay? What do you think? What if we use CRISPR to cure diseases? What if we use it for other purposes, like to make smarter or stronger people? What if only some families can afford it? It is important that we ask each other these questions and that we include concerned individuals from all over the globe. That way, we can choose the path that better takes into account the needs and worries of everyone involved. And we're done! Congratulations! What did you think? Did you learn anything? Did you ever think we could learn how to edit genes by studying how bacteria fight viruses? Did you think we should edit genes? Are you going to go study the vocabulary for this article now? Cool. Also, if you found that text a little difficult, but not too difficult, try reading it with me again and on your own a few times. If it was too difficult, put this text aside and check the list down below for some suggestions of easier text read-alongs to try before you eventually come back to this one. If you found it too easy, you can check the description below for some suggestions of harder text read-alongs to try. Great job! See you next time.